Hello guys, welcome to our channel. Do you hear the word DNA library? Yeah, it's true. In this video, I explain what is it and what are the uses of that DNA library. Now we came to the topic. We know a typical library has a collection of books, unlike the DNA library has a collection of genes of an organism. Hence, it is called DNA library. These genes are stored in vectors DNA. It is also called the genomic library or gene library. In this slide, the genomic DNA library can be compared to the classical book library. In a classical library, books are stored, and while in the DNA library, DNA fragments are stored. A book is safeguarded by its cover, while the DNA is safeguarded by the vector DNA. A book is stored in a bookshelf, while a DNA fragment is stored in a host cell in culture flask. Many books are stored in bookshelf, while several culture flasks are stored in culture collection. In the book library, books are replicated by xeroxing, while in DNA library, DNAs are replicated by subculturing the host cell. Now I explain in simple way. The DNA is considered to be a pages of book. The DNA is attached to the plasmid. Now the plasmid is compared to the book cover, and then the plasmid is inserted into the host cell. Now the host cell considered to be a book. The books are arranged in a bookshelf in typical library. In DNA library, the host cells are arranged in culture flask. The culture flasks are arranged in culture collection. In the book library, the books are replicating by xeroxing, while the DNA library, the DNAs are replicated by the replicating process of the host cell. And the main purpose of the creating genomic DNA library is to arrange all DNA fragments of a genome in a proper order and to supply the required DNA fragments to researchers for further studies. And next, vectors used in DNA library. There are different kinds of vectors are used in DNA library. They are plasmids, cosmids, lambda phage, PAC, BAC and YAC. Why those kind of vectors are used? Because each vector has an ability to accommodate different length of a fragment. So the length of the fragment is choose the vector. And now we will know about those vectors one by one. Plasmids can accommodate less than 15 kp long DNA fragment. And next, the cosmids can accommodate 30 to 45 kp long DNA segments. And the lambda phage vectors can carry 10 to 25 kp long fragments. And the PAC can accommodate 70 to 19 kp long DNAs, and a BAC can carry 100 to 500 kp long DNA fragments, and the last YAC can accommodate 250 to 1000 kp long DNA fragments. YAC can hold long DNA fragments, and this method is used in widely. And these are the vectors used in DNA library. And now we know about the uses of genomic library. Why the genomic library is important? Let me know. To learn the structure and function of a genome, and to map the genome, and to identify the specific gene, and to diagnose genetic diseases and find out the genetic variations between the species, to find out evolutionary relationships, and to multiply a desired gene to create a genetic modifications. These are all the uses of genomic library. And the next class we will learn about the creation of genomic library and the screening of DNA library. Thank you guys. If you found this video is helpful, like, share and subscribe it. Thank you.